So this is what I consider probably one of the most important sessions of this meeting, which is the presentations by some of the younger scientists that represent the future of synthetic biology. So we should pay close attention to what they have to say. And our first speaker is Julius Lux, who is from Adam Arkin's lab at UC Berkeley. Thank you.
Okay, so thanks a lot. I'm Julius Lux. I'm a Miller Fellow in Adam Arkin's lab at UC Berkeley, and we're also part of this Synthetic Biology Engineering Research Center, the NSF program there. And what I want to tell you about is some of our early work on engineering a transcription control system in bacteria that uses antisense RNA. So let me give you a little motivation. Why do we want to do this? Why aren't we just satisfied with using proteins to control transcription? There's a lot of reasons to use RNA, and one of the good reasons is we already have a lot of elements of control of gene expression in other RNA components. So, for example, we know about how to make secondary structures and put them in the upstream region of genes that can control strand stability. We know about some regions that you can put downstream of the genes. Christina Smolke's done a lot of work on putting ribozymes down there that when they cleave also control transcript stability. You can design secondary structures to sequester ribosome binding sites and open those up and thereby gain a little bit of control on translation. And so one of the things we're interested in is trying to put the transcription piece. All this stuff basically works with RNA only. We want to add in the transcriptional piece. So then you really have all the degrees of freedom and control of gene expression on the RNA level. Another good thing about RNA, and part of the reason why some of this has been so successful, is that we have a clue about RNA designability. So Watson-Crick base pairing rules get you pretty far when you try to design some of these things. And using those this designability property, you can really start to think about RNA parts that have certain key properties, and these are the parts that are going to really propel us to the next level of building genetic circuits inside cells, what a lot of people want to do. So some of these properties are part independence. This is clearly an important thing. If you want to pull two parts off the shelf and have them work in the cell, they have to function independently of each other. And so this is an example. If you have one transcript over here controlled by these elements, it's usually not going to interfere with this transcript over here. So you already have a good degree of independence. The RNA is tunable. Like I said, Watson-Crick base pairing rules. You can play around with the base sequence here. Maybe you want to bind up this RBS stronger. You can tune not only strength of interaction, but also specificity. And I'll get to more of that later in this talk. Further along the tunability, you have orthogonality. So this is where you'd have two parts that have the same function, but you tune them so that they don't interfere with each other. So this is like orthogonal repressors or something like that. You can play the same games with RNA, and clearly you'd want to do that to have a lot of parts to play around with to build these circuits, larger circuits. And then composability is a real key property here. So in this case, I had two different elements, each performing a distinct function on the same physical molecule. And that allows us to hopefully predictably combine functions together to create new functions and build circuits out of. So kind of the confluence of all these four properties really lead to scalable parts families. These are parts that you could really, truly, well, hopefully, in a predictable way, pull them off the shelf and put them together to build bigger and bigger circuits. I think as a field, we need to start focusing on trying to find parts that have these properties so we can start to realize some of our dreams. So like I said, we're also interested in the transcription control area. And I should mention that a lot of this great work is done by Christina Smolke has targeted a couple of these regions, and another reason why we want to do transcription is so we get to have some fun too. Okay, so I'm starting to tell you about some of the details of this. So we're starting from a natural control system that's coming out of this plasmid named PT181 that actually controls its copy number through an antisense RNA-mediated interaction. And what happens is there's a specific gene in PT181 called RepC. When RepC is made, it binds to the origin and causes plasmid replication. And the mechanism that the plasmid uses to control its copy number involves about a 200 200 base pair sequence upstream of this gene. And this has been studied enough to know that after a polymerase has transcribed about 130 base pairs of this region, this thing is known to fold in the specific secondary structure that has these 3 hairpins like this. Now, if this process continues in the presence of a piece of antisense RNA, and this piece of antisense RNA is antisense to these 2 front hairpins, what happens is the free base pairs in these loops of this antisense piece and the loops of the leading strand can interact with each other since they're free. They initially form a kissing hairpin structure, and then it further is allowed to form a duplex. And what this duplex allows is the formation of a downstream transcriptional terminator I've highlighted here in red. And when that terminator forms, polymerase is kicked off and you don't get any of that gene product. Now, if antisense is not present, what happens is that the rest of the sequence down here causes a refolding of this structure. And that refolding actually sequesters the terminator. It allows transcription and you get gene expression. So that's basically the natural system. And what we wanted to do is play around with it and see how, if we can change this system to manipulate it, start to get at some of these properties of parts that I talked about earlier. Specifically, what I'm telling you about today is initial work on looking at specificity, so the tuning aspect, particularly since the initial interaction between the antisense and the leader sequence here occurs between this kissing hairpin interaction. Our hypothesis is that if we mutate this region, maybe we can get to some orthogonality, or maybe we can improve the performance of this device. And the other thing I want to tell you about is composability. Can you put 2 of these together? What happens if you put 2 of these together? Does it work at all? What is its function like? So briefly, I'll tell you about how the wild type function performs, and basically tell you about our simple experiment. It's just a fluorescent assay. So we do have a construct, have this leader sequence up here, and we replace the natural gene by GFP. And without antisense, you get a large GFP signal. And then we add in antisense, and then the antisense causes this terminator to form, and you get a lower GFP signal. And the data that I'm going to show you is going to be composed. I'm going to tell you about what we call transcription attenuation factor, which is basically the big one minus the small one divided by the big one. So it's kind of the percent that you've attenuated the transcription here. So in particular, we have a 2 plasmid system. This GFP construct is on a medium copy number plasmid, and the antisense is inducibly expressed from PLAC on a high copy number plasmid. When we do this and we induce PLAC, we get an attenuation factor of about 60%, which is consistent with what's in the literature. So that's what the wild type does. And one of our goals is to try to improve this, or try to figure out how to improve it, really. And one of the things we could do is we could build a library of these things and test them out and all that stuff. But what we decided to do, since we know a little bit about RNA secondary structure prediction, is to try to do it computationally first. And our motivation for that was some work done by Brantel and Wagner, where they did a series of RNase digests and mapped the digest onto structures predicted by the MFold program for this particular sequence. And what they found is that the structure predicted by MFold is actually consistent with the RNase digest maps. So that led us to think that, it's not going to get us 100% there the way, but maybe a computational study would help us identify good mutants to build in the lab and then test. Okay, so what did we do computationally? Like I said, we were going to target these two loop regions because those are the ones that initially interact. And our computational workflow, so initially when I started out, I was like, oh, this is not going to be hard. I'll just change the wild type sequence here to like GGG and CCC and make the interaction stronger, and that'll be totally fine. So then you type it into the computer and you get it to fold, and actually this thing folds completely differently. And one of our hypotheses is that this mechanism for it to work, for it to function correctly, absolutely requires this particular secondary structure. So I was a little bit disappointed, and then I tried different variations of GGG and CCC or whatever. But after about 5 or 6 of them completely failing, I decided that we should just, generate them all on the computer, get the computer to fold them all, and see what we get. So that's what we do. We have this leading strand here and the antisense strand, and these are the 2 regions represented schematically here. And what we're going to do is assess all possible mutations in these 6 base pair regions on the 2 loops. So the first part of the computation is just generate all possible mutants. And I should say that we do them separately at first. So I generate a bunch of mutants that only involve mutations in the left region, and then I have a separate list of mutants that have mutations in the right region. And when I say mutant here, I mean both mutant sense and its cognate antisense strand here. So after you get thousands of mutants here, then we do a reduced step where we basically take the list of mutants and filter out the ones that are going to be predicted to be bad. And initially we had 3 hypotheses. I mentioned that we needed the secondary structure to be correct. So at this first level, we filter out all mutants that have an incorrect fold in the leading strand. At the second level, we filter out all the mutants that have incorrect fold on the antisense strand. And then the third filter we apply here is based on an estimation of the free energy of interaction between these 2 hairpins. And our hypothesis is that if you have a stronger interaction here, you're going to have a better attenuation factor. So we only kept mutants that had a predicted estimated free energy of kissing interaction lower than that of the wild type. So at this point in time, for the left-hand side, only about 3% of all the possible mutants had survived this computational screening procedure. And after this, we just took the 50 mutants from each pool, crossed them together. So now these mutants in the gray box represent mutations in both loops, and perform the same filter to make sure that everything folded correctly and whatnot. Okay, so now basically at this stage I have a list of mutants, a bunch of them, and we're interested in specificity. So what we did with these now is took this estimated free energy of kissing interaction and computed a matrix of this energy. So in this matrix, blue means a low free energy of interaction, and red means high. And what we've done on the ith axis here, that represents the ith mutant leader sequence, and on the J column is the antisense. So this dot represents taking mutant antisense 2000 and hypothetically reacting it with mutant sense 100 or whatever. So zooming in on this a little bit more, we've actually placed the wild type here for reference in the 0, 0 coordinate. So all of our mutants are predicted to have a more stable kissing structure than the wild type, which is what we hope they would. That's a part of our filter requirement. And then we zoom in even more, just look at this little box, you see that, okay, this might be a good candidate for a mutant to build and test out if our hypotheses are correct, because these off-diagonal terms are red, meaning hopefully that if I put the wild type leader sequence in front of GFP and try to express my mutant antisense, nothing would happen. And this is good because eventually you want to control many genes at once with these things and not have the controlling machinery interfere with each other. So our preliminary experiments for this show that, so these are them. I'm plotting the data in 2 different ways. This is just a typical plot, and here is basically the same box here with the same color scale. So the first bar shows you the wild type, 60% transcription attenuation factor that I showed you earlier. This bar shows you the mutant. So you can see it works. It's 40%. It's not what we had hoped, which would be better than the wild type. But it's a noticeable effect. And the off-diagonal terms are significantly lower than these 2. Although ideally, you'd want this bar to be really high and these 2 to be really low to have true orthogonal parts. But what this really told us was that we're kind of on the right direction with this, and we're doing a bunch more experiments now to really try to figure out if our initial computational hypotheses were correct. Where are they wrong? Do we need to add new filters to the program? Okay. The other thing I want to tell you about is composability. So what happens when what we want to do is put 2 control regions in series together up front of one gene. And so I told you earlier that just by changing a few base pairs in one of these loop regions, you can completely control the RNA's secondary structure. So there's almost no reason to think that this would actually work because RNA folding is such a global phenomenon. But when you pop this into one of these secondary structure prediction programs with a specially chosen insulator sequence in between them, you can see from this diagram, it's not really drawn very good, but you can see basically that these 2 regions are folded the same. And what that represents is that the 2 regions look like they might fold independently of one another, which is good. Maybe they won't interfere with each other and they form a composable part here. So when we do the experiments, this is what we get with a single attenuator, about 60%. And we add 2, we get 80%. So we've increased our effect. And this is completely consistent with what you would predict if you multiplied basically squared the single attenuation efficiency. And that makes sense mechanistically because as the polymerase is moving along, only 40% makes it through here, and then 40% of that 40% is going to make it past here. So the multiplication is what you get when you have when these parts are composed together. So as another test with that, we can actually play with that mutant that I showed you and build different possibilities. So instead of just two wild-type attenuation systems in front of each other, I can put a wild-type, then a mutant, or a mutant, then a wild-type, or a mutant, then a mutant. And if this multiplicative property is really true, then I should be able to take my experimental results from my single experiments on just the wild-type and just the mutant, combine them together to get what I would see in these combinations. So when I try each of these four things versus the wild-type antisense strand, you get the following thing. So what I'm plotting here is attenuation factor. In blue is what you would predict from this simple equation, and in yellow is what you get in the experiment. And you can see pretty much for everyone within error bars, and these are actually pretty close. It really looks like the multiplication thing is right on. And I can do the same thing by using the mutant antisense strand here, and it's the same thing here. So not only does composability is really interesting because it gets us a route towards boosting the attenuation factor, but it looks like it's really predictable. And if we got our mutants to perform better themselves, this is the type of effect that you could use to build logics in these systems. Okay, so just to wrap up, I told you a little bit about our transcription attenuation scheme with RNA and about some of our early work on looking at specificity, seeing we can mutate these regions here to design potential, the goal here would be to have a large orthogonal family of these parts to use for transcription control. And then our composability experiments, playing around with how you can put these things together. Special thanks for Sinberg and the Miller Institute for funding. Stanley Shi, unfortunately, couldn't be here today, but he's done a lot of this work with me, and he's a really excellent grad student in Adam's lab. Sarah Richardson was a Krell Fellow from Johns Hopkins. She spent the summer with us and really helped us out a lot. And Adam Arkin, who is a fantastic guy to work with. Thanks a lot.
Thank you. Are there any questions? Yes.
Yeah, so I'm curious in terms of how many of your 2,000 combinations you tested and whether you can start to correlate success with some of the multi-criteria that you've used initially and see whether you can re-weight those to have a better predictive algorithm in terms of what's going to work.
Yeah, no, exactly. So I thought that what we've been trying up till now is like running through these very specific hypothesis-driven. So we've done some other experiments where we've tweaked our things and tried to build another mutant. But it's definitely clear that right now what we should do is go ahead and build a bunch of these things, see which ones work, and try to statistically back out the right properties. Definitely the thing to do, I think.
So everything what you did is very specific for this kissing structure.
Right.
And you never tried simply take antisense RNA of 20 or 50 long, which is really antisense, and see that image.
No, we have not tried those for the transcription. And
Systematically, without kissing.
yeah, no, I mean it could be. Yeah, but the other thing you got to be really careful about. You can't just take 20 to 30 nucleotides and hope that it would bind, because they're going to also fold into secondary structures and things like that. So initially, even though this thing is perfectly antisense, it comes through with this kissing interaction. So there might be sequences like that would preclude full duplex formation and things like that. But it'd be very interesting to examine.
As we talked before, we tried to do just that. We make a random differentiation.
Oh.
We just tried to take any RNA and run from another plasmid or in different combination, and we never get more than 30% inhibition and mainly nothing.
Yeah.
So you have to design.
Yeah.
Okay, lots of experience there.
Yes.
Are there any other questions? Okay, fabulous. Thank you.